Hi everyone, I'm great pleasure to welcome you to another talk in series of Global IMMUNOTALK. My name is Kazuyo Moro from Osaka University Graduate School of Medicine and Riken IMS Japan. Just before I introduce today's speaker, I would like to remind you next week's speaker is Juliana Itoyaga from Stanford University. Now on behalf of all organizers of Global Women Talks, I would like to introduce today's speaker, Shimon Sakaguchi, who is an immunologist recognized particularly for his discovery of regulatory T cells. So Shimon is a distinguished professor at the Immunology Frontier Research Center, IFREC, Osaka University, Japan. And Shimon obtained his MD in 1976 and PhD in 1982 from Kyoto University, beautiful city in Japan. And after finishing postdoctoral study at Johns Hopkins University and also at Stanford University, he became assistant professor in the Department of Immunology at the Scripps Research Institute. And he returned to Japan in 1991 and continued his research at the head of Department of Immunopathology at Tokyo Metropolitan Institute of Gerontology in Tokyo. And from 1998 to 2011, he was a professor and also a director for several years at Kyoto University. And finally, in 2011, his lab moved to Osaka University, and since then, he is a member of IFREC Osaka University. So he has won so many hours, and if I introduce all of them, I might run out his time to speak. So let me introduce a few of them. So for example, he has received the Gardner Institute International Awards in 2015, and also Robert Koch Prize in 2020. And Order of Culture in 2019 from the Emperor of Japan. This is very big prize in Japan. So the younger generation viewing this Global IMMUNOTALK may not have heard about immunosuppressive T cell, but I heard that there were so many discussions about the existence of immunosuppressive T cell until Shimon discovered regulatory T cell. So he investigated the property of T cells with autoimmune suppressive activity in mice. And in 1995, he demonstrated that the depletion of T cell subpopulation leads to the spontaneous development of the variety of autoimmune disease. And discovery of the transcription factor FOXP3, which is essential for Treg cell development and function, is also his great achievement. This discovery revealed that the mutation in the FOXP3 causes autoimmune disease, allergy, and also inflammatory bowel disease, and the Treg are essential for immune cell tolerance and homeostasis, even in the human. So Shimon has been studying Tregs for almost 40 years since then, and I expected that today he will talk about Tregs, including their potential as a therapeutic target. So now, Shimon, could you please turn on your camera for us? Okay, great. And first of all, thank you very much for joining to this Global Immune Talks. And we're really looking forward to listening to your talk today. But since we want to know a little bit more about you, we would like to ask you one question. So the question we would like to know is, how would you describe your personal style, and how did it impact your scientific career? So,
All right. Well, first of all, thank you, Kazuyo, for a kind introduction. It's my pleasure to join this Immune Global Talks. Yes. Well, so about your question, I think it's important to have the skill of being insensitive. Well, it may sound odd, but nowadays, lots of information available for a scientist. If the paper is published, immediately it is available through internet. So what is important is to concentrate on what you think is important. So for that, being insensitive, well, sometimes you must shut the door to the information from outside. That is important, well, for scientists. Well, actually, I read one article, and where André Leboeuf, he's a Nobel laureate, a French scientist. He said, what is lousy to scientists is papers by others. So that is kind of, in a sense, it's true. So, well, to have the skill of being insensitive to insensitive anyway, and concentrate on your work. That is, I think, especially nowadays, it's important.
Thank you very much for sharing that story. I think I also need to be insensitive in the future. Okay, so now, if you are ready, could you please share your slide for today's talk and start your presentation?
Mm-hmm. Just a moment.
Yes, it's great. Looks fine.
Okay.
Okay.
Well, so today I'd like to talk about Treg, regulatory T cells, and their roles in immune tolerance. So outline of my talk is the following. First, let me talk about a brief history of Treg research and the molecular basis of Treg development and function. And second, a human Treg, how they contribute to autoimmune disease, the monogenic and the polygenic ones. And third, and then based on these findings, how we can develop a Treg-based therapy of autoimmune disease, especially antigen-specific immune suppression, which is still is a holy grail in immunology, antigen-specific suppression. So let me talk about history. As shown on this slide, it is estimated that around 5%, approximately 5% of the population is afflicted with some sort of autoimmune disease. So here, as the organ-specific autoimmune diseases, and then systemic ones. And then, so I'll use some autoimmune diseases as a readout of our experiment, especially autoimmune gastritis or thyroiditis, type 1 diabetes, as a breakdown of self-tolerance. So one feature I think is important, that is the overlapping of autoimmune disease in the patient. So if you find a patient with type 1 diabetes, he or she may also have thyroiditis or gastritis or other autoimmune diseases in humans. This is also the case in animals. For example, NOD mice, which develop type 1 diabetes. It's a good model of type 1 diabetes. But when they become aged, they show thyroiditis. And BB rats, which also spontaneously develop type 1 diabetes. But at the same time, they have very frequently thyroiditis and gastritis. And also a chicken, obese strain chicken, which spontaneously develop thyroiditis. But then they have also other autoimmunities, including gastritis. So these findings indicate that, well, they raise the questions. So is autoimmune disease due to anomaly of the immune system or target self-antigens? So immunologists use EAE, experimental allergic encephalitis, as a model of autoimmune disease, multiple sclerosis. So immunizing mice or rats with the self-antigen plus a strong adjuvant, complete adjuvant. So postulating, it postulates that somehow something happens at the target antigen. But in these findings in humans overlapping, it suggests that, well, whether this is really autoimmune disease due to anomaly of self-antigens or the immune system. So that is, I think it's an important question. And second, so based on these findings in mice, rats, and chicken, and humans, so is tolerance mechanisms and preventing autoimmune disease, is it evolutionarily conserved? To address these questions, I think there are two important experimental autoimmune diseases, which is induced by manipulating the immune system, not self-antigens. So one is in mice. If you thymectomize, removing the thymus around day 3 after birth, within two months, some of them have developed various autoimmune diseases, say autoimmune gastritis, ophlitis, that is autoimmune disease in ovaries, thyroiditis, and etc. So this finding was made by Japanese pathologist Yasuaki Nishizuka and Teruo Sakakura in 1969. So this is a very nice model to analyze self-tolerance. The second model is in rats. So normal rats, when they were thymectomized around 6 weeks of age, and after that, every 2 weeks, when they are irradiated to gray, 200 Röntgen, every 2 weeks, 4 times, and then some of them developed autoimmune thyroiditis and others type 1 diabetes. So anyway, just manipulating the immune system, we can induce autoimmune disease in normal animals. So this is a post-thymectomy autoimmune disease. Normal bulb C mice, for example, day 3 thymectomy can induce gastritis 30%, ophlitis around 30%, but at day 0 thymectomy, within 24 hours after birth, almost nothing happened. Day 7, nothing happens. So a critical day of thymectomy, that is important. And also, if you change the genetic background of the mice from bulb C to A strain, and then you see 90% develop the ophlitis and 20% orchitis, autoimmune disease in testes, gastritis, and thyroiditis. In contrast, C57 black mice, this strain is very resistant. Almost nothing happened by day 3 thymectomy. So the day of thymectomy is critical, and also genetic background is important for what kind of autoimmune disease develop or not. So this, the important findings, and one important finding in this post-thymectomy autoimmune disease is that if you inoculate normal T cells from adult mice, non-thymectomized adult mice, before or after thymectomy, you can prevent autoimmune disease. So as shown in the previous slide, by using A strain, and then thymectomy, day 3 thymectomy can induce ophlitis, about the 90% of the mice. So day 3 thymectomy can induce autoimmunity. But if you inoculate 5 million T cells before or after 1 week after or 2 weeks after, you can completely prevent disease development. Then 3 weeks or 4 weeks later, now the mice become bigger, and then the 5 million T cells not enough to prevent disease. So how we can interpret these findings? So thymus producing T cells from gestational days on day 0, the pups are born. And then day 3 thymectomy can induce autoimmunity. This means that the self-reactive or autoimmune T cells, are pathogenic ones, they are already produced by the thymus, and they have migrated to the periphery before the day of thymectomy. And then around day 3, perhaps the sort of suppressive, autoimmune suppressive T cells, they are produced and then migrate to the periphery. So if you remove the thymus around day 3, the autoimmune T cells, which have already peripheralized, migrated to the periphery, they become spontaneously activated and cause autoimmunity. And if you inoculate normal, perhaps immunosuppressive T cells, you can prevent the disease. Oops. Let me see. So to verify this mechanism, what is important is not by thymectomy, but more directly, if you define regulatory T cells and autoimmune T cells, and then directly remove this population regulatory T cells from the normal adult mice, and that is the best the demonstration of this kind of the T cell regulation in autoimmunity. So what we and others did in the past is on this slide. So here normal biopsy mice. If you take out spleen T cell suspensions, and then you remove certain population of T cells by the expression of cell surface molecules, and then transfer the remaining cells into T cell deficient nude mice. If we can successfully remove the suppressive T cells, they may spontaneously develop various autoimmune diseases. So this was indeed the case. Many years ago, we first removed the CD5 high cells, and we could induce various autoimmune diseases. And 5 years later, Fiona Powley and Dan Mason at Oxford University, UK, they showed that if you remove the CD45RC or RB low population, and then transfer, they use the nude rats, not nude mice, but then they found that they spontaneously develop type 1 diabetes and systemic autoimmunity. Then the question was, any more specific markers for this suppressive population? More specific means by removing the smaller number of cells, and then if you can induce broader spectrum of autoimmune diseases with more severe pathology, and that means that marker is more specific. And then we tried many various molecules, and then came up with CD25. And CD25, the 10% of CD4 positive cells, they express CD25, IL-2 receptor alpha chain, and less than 1% in CD8 population. And then CD25 positive cells, they are CD5 high, and then CD45RC low. So it's a good candidate. And then indeed, if you remove 10% of CD4 population expressing CD25, we can induce various autoimmune diseases and also inflammatory bowel disease. So this is autoimmune gastritis. So the gastric mucosa is destroyed by infiltration of inflammatory cells. And then you can demonstrate autoantibodies against gastric parietal cells. And this is actually the proton pump required for gastric acid secretion, like in human autoimmune gastritis with pernicious anemia, thyroiditis with antithyroglobulin autoantibodies, and insulitis with type 1 diabetes. So this mouse developed type 1 diabetes, and then the disease can be transferred to other mice by T cells. So CD25 positive cells, they looked like the immune suppressive population. And if you look at the ontogeny, day 3 in the spleen, you'll see CD3 positive cells, but not a CD25 positive population. Day 7, some, and at day 60, around 10% of CD4 population, they are expressing CD25. So individual mice in the spleen, the percentage of CD25 positive cells among T cells. So after day 3, it's rapidly increased to the level in adult mice, around 10%. So day 3 thymectomy, perhaps they abrogated the peripheralization, peripheral migration of this population and cause autoimmune disease. So neonatal day 3 in mice. So this is, well, why day 3, you may ask. But in mice, it's day 3, but in rats, it is day 0, especially within 12 hours after birth. And humans, around 12 weeks in utero, in gestation. So mice and rats, they are born in immature state. Their ears and eyes still closed compared with humans. So these findings raise a question. So is autoimmune disease due to developmental or functional anomalies of T cell subpopulation? So if you are working on immune tolerance and autoimmune disease, we may talk about deletion or allergy or suppression or whatever. But from a developmental immunology aspect, the really anomaly of the one population, T cell population, its developmental anomaly or functional anomaly can be causative of autoimmune disease. That is, for us, that was at that time the exciting finding. So here, normal thymus can produce, of course, naive T cells, and then also the Treg regulatory T cells can be selected by recognizing the same self-peptide MSC. But the Treg, one cardinal feature of Treg, natural Treg, is that they differentiate and become functionally mature already in the thymus, and then they migrate to the periphery. So this zigzag line means it takes time for them to become mature functionally. So there is time lag in leaving the thymus compared with naive T cells, a bit delayed. So that is the reason why the thymectomy, day 3 thymectomy, can somehow relatively reduce this Treg population and allowing the development of autoimmune disease. Then the question is, whatever it is, genetic anomalies or environmental insults, if they affect the balance between Treg and then effector T cells or autoimmune T cells, they may be a causative of autoimmune diseases. So as already I have talked about the X-ray ionizing radiation. They are Tregs are highly proliferative. So perhaps one of the most proliferative population in the circulation in the blood, that is regulatory T cells. They are always they proliferating, and then they are dying. So ionizing radiation, they are sensitive to ionizing radiation. And then CD25, this is the IL-2 receptor alpha chain. It is important for Treg function. And cyclosporine A, if you by inhibiting IL-2 production, then Treg will die and you'll see autoimmunity. And certain viruses, which they selectively infect the proliferating T cells. For example, HIV, AIDS virus, they Tregs are more susceptible to that kind of retroviruses. So the environmental, biological or chemical, physical, environmental insult can cause autoimmunity. Now, how about genetic ones? And so it is already known that the CTLA4 or CD25 or CD122, if you make these knockout mice, you'll see a very severe autoimmunity. I'll come back to these later on. And the most important one is FOXP3. So around 1982, the disease called IPIC syndrome was reported in a pediatrics journal. So the important features of IPIC syndrome is that more than 80% of the patients develop type 1 diabetes within 3 years after birth, more than 70% thyroiditis, and also other autoimmune diseases, say hemolytic anemia, and then also inflammatory bowel disease, almost 100%, and also hyper IgE and allergy. So when they start milk feeding from the mother, and then the intestinal the microbes, commensal microbe, they develop, and then immediately they show the inflammatory bowel disease, which is hyperreaction to commensal bacteria in the intestine. And then scurvy mice, it is a counterpart in mice. It has a mutation on X chromosome. The first, the disease gene was positionally cloned in scurvy mice, and immediately it was found that gene was responsible for IPIC syndrome. So that gene, FOXP3 transcription factor. And then, as shown here, 10% of the wild mice express FOXP3, but the scurvy mice, they don't. So the IPIC syndrome, it's very similar to what we saw after removing Treg in normal mice. So we wondered what's the relationship between Treg and the FOXP3. So as shown here, if you stay in normal spleen or thymocyte for FOXP3 and the CD25, the majority FOXP3 positive cells in the spleen, they are expressing CD25 and also in the thymus. So previously we removed this population. That means we removed the majority of FOXP3 positive cells. So we could induce autoimmune disease. So now in human and the mice, the CD25 on the cell surface and FOXP3 in the nucleus, that is the best marker for defining regulatory T cells. So the important function of FOXP3 is that it can confer a suppressive activity on conventional T cells. So just retroviral transfection expression of FOXP3, and then you can convert the conventional T cells into Treg. So FOXP3 transduced cells, they can prevent the colitis or autoimmune gastritis when they were co-transferred to nude mice. So normal colon, colitis can be prevented. And normal gastric mucosa, gastritis, and gastritis can be prevented by FOXP3 expressing conventional T cells. So these findings all together, what these findings suggest to us is that the normal individuals, they do possess the potentially hazardous self-reactive T cells, but their activation and expansion is somehow controlled by regulatory T cells. And if you remove regulatory T cells, some common autoimmune diseases, such as type 1 diabetes and thyroiditis, they spontaneously develop. And at the same time, the immune responses against, say, conventional the bacteria or viruses, it's also enhanced. So the immune responses, now hyper responses to commensal bacteria in the intestine, it causes IBD, inflammatory bowel disease, and allergy. So this means that the border between self and non-self is not fixed. You can move this upward or downwards. If it's downward, autoimmune disease may happen, but also you can evoke a good anti-tumor immune responses. And by raising this threshold, you can induce the transplantation tolerance. So here, the tumor immunity is the same experimental system, nude mice are transferred with CD25 minus spleen cells. And if you inoculate syngeneic tumor cells, and then the tumor become bigger and then rejected. After rejection, the 10 times number of tumor cells, then immediately rejected, meaning that these mice become immune to this particular the cancer cells. So without removing Treg, CD25 positive cells, the tumor become bigger, and then mice die of tumor progression. And the 1-to-1 ratio mixture, Treg become half, but still suppress immune responses. So tumor become the bigger. And then only this population, this group can survive by rejecting tumors. And transplantation, again, nude mice. So they can accept allogenic skin because they don't have T cells. And then, so if you transfer T cells alone, and then within a month, all rejected. But if you transfer first Treg, and then within a week, they become sensitized and then proliferate, sensitized to alloantigen, and then recruited to the graft site and suppress the immune response when the T cells are transferred. So here, 1-to-1 ratio, they now 30% become permanently tolerant. It should increase Treg, say 1-to-3 ratio, and then 70% become permanently tolerant. So without immunosuppressant, just expanding the naturally present Treg, you can induce a transplantation tolerance. So these findings all together, now we know that the Treg, not only autoimmunity, or tumor immunity or transplantation, they are involved or they play key roles in the fetal-maternal tolerance or immunometabolic disease and even the tissue repair. So Treg produced by the thymus, we now call a thymus-derived Treg, T-Treg. And then some of them, especially in the intestine, in the intestinal mucosa, naive T cells can differentiate into FOXP3 positive cells. They are called peripherally derived Treg, P-Treg. And also in vitro, in the presence of TGF-beta, IL-2, and antigen expiration, naive T cells can be induced to become FOXP3 positive. They are called induced Treg, iTregs. So now let me talk about how Treg, about the Treg function. And so FOXP3, as I mentioned, if you transduce FOXP3 in conventional T cells, you can confer suppressive activity. So FOXP3 controls the hundreds of genes and downstream genes. And then which one is important? Well, and so we think that the downstream gene, if the gene is the knocked out or over the expression, and then you'll see, if you see autoimmune diseases similar to those found in FOXP3 mutation or deficiency, that gene is important. And then from that point of view, not so many knockout mice, gene knockout mice with severe autoimmunity. Of course, genes must be controlled by FOXP3. So one group is IL-2 related, CD25, IL-2 receptor alpha chain, CD122 beta chain, and IL-2 itself. These knockout mice, they spontaneously develop various autoimmune diseases. Indeed, FOXP3 upregulates CD25, but it represses IL-2 production. And another gene is CTLA4. So regarding IL-2 and IL-2 receptor, I think, well, a cardinal feature of natural Treg, that is, they are expressing the high affinity IL-2 receptor, alpha, beta, gamma, already in the thymus, and then expressing constitutively. So that is a difference from activated conventional T cells. So conventional T cells, only they express the high affinity IL-2 receptor only when they are activated. But the Treg, when they are produced by the thymus already and constitutively, continuously they express the high affinity IL-2 receptor, and they don't produce IL-2. So because of a FOXP3 repression by FOXP3. So this means that they are highly dependent on exogenous IL-2 for their survival. So if you knock out these IL-2 receptor chains or IL-2 production, then Treg cannot survive. So here, just to demonstrate this, just one injection of anti-IL-2 neutralizing antibody, and then you see the several days later, in the thymus, also in the periphery, the Tregs they are gone, they die. So by just neutralizing IL-2, you can induce autoimmunity. So just an IL-2 neutralizing antibody twice into normal mice, normal bulb C mice, you can induce the autoimmune gastritis with high titer of anti-parietal cell autoantibodies. So this is NOD mice. So they have an insulitis. By IL-2 neutralization, and then insulitis become more severe. And thyroiditis now develop very severe thyroiditis. And also in other organs, lacrimal gland or salivary gland, the autoimmune salivary gland adenitis. How about CTLA4? So Treg, the specific positive cells, they are constitutively expressing CTLA4 already in the thymus. And you make a CTLA4 conditional knockout mice, Treg specific ones. And then conventional T cells are activated ones, and they express CTLA4, but in Treg, they don't. And then these mice spontaneously develop the various severe autoimmunity, for example, myocarditis. So this is atrium, and then you see the cardiac myocytes, they are destroyed with inflammatory cell infiltration. These blue parts, the ventricle, they are destroyed by autoimmune and aggression. And so they show the cardiomegaly because of heart failure. And then in the circulation, IgE concentration several hundredfold higher, and then tumor immunity. So they'll die soon. So if you can transfer spleen cells into nude mice and then false spleen cells, and then inoculate the tumor cells. You see red line knockout mice, they reject the tumor cells. So they develop autoimmunity, allergy, and also strong tumor immunity, as shown in Treg depleted mice. Again, just anti-CTLA-4 antibody, just twice into normal bulk C mice, you can induce severe autoimmune gastritis. So this is the historical, very historically severe. So anti-CTLA-4 antibody, ipilimumab, now it is in use, clinical use in tumor cancer as cancer immunotherapy. But the side effect is one of the side effects is autoimmunity, as shown here. And then these not a conditional knockout Treg, they are impaired in vitro suppression. So this is allogeneic MLR, and then the normal Treg, blue one, they can suppress the allogeneic T cell responses, 1:1, 1:3 ratio. But the red one and conditional knockout Treg, they can't. How then the CTLA-4 contribute to Treg suppression? So here, this is the in vitro culture, APCs, and then this is Treg, and then CD86 green, and CTLA-4 red. And what you see is that by stimulating Treg by anti-CD3, and then they interact with APC, and then the CD86 become aggregated, CTLA-4 become aggregated. And then they are transferred from APC to Treg. It is called trogocytosis. And some of them become further endocytosed. It is called transendocytosis. So, and then what happened to APCs? So this is dendritic cell, splenic disease, and co-cultured with Treg. So they are the majority of the expressing a high level of CD80. And then if you co-culture with CTLA-4 knockout Treg, and not much change. But you co-culture with wild type Treg and with TCR activation. Then the CD80 high, mature DC, it becomes smaller in population. In contrast, free PD-L1 high, and then CD80 low population, now they develop. And then with tailless Treg, tailless means the cytoplasmic portion of CTLA-4 is truncated. So they no signal transduction to Treg through CTLA-4, or no endocytosis. And then this population, free PD-L1 high, CD80 low population, now they expand. So what this means is that now it is known that in normal APCs, CD80 and PD-L1 is cis-associated. And if Treg removes CD80, then the free PD-L1 increase on the cell surface. And only free PD-L1 can interact with PD-1 positive effector T cells. So this means that the Treg remove CD80-86 and also upregulate free PD-L1. So the Treg can suppress naive T cells by reducing CD80-86 the expression, and also inhibit effector function of PD-1 positive T cells by increasing free PD-L1. And then what happened to the T cells under these Treg-mediated suppression? So this is a human peripheral blood, and then you can isolate CD8 positive cells and Treg. So CD8 positive cell alone, and then stimulated with melang A or MALT1. And this is the self-antigen, the target antigen of vitiligo vulgaris, and also tumor antigen in melanoma. So without Treg, and then you can detect antigen melang A specific T cells by tetramer, and then you see here that without Treg, now 1 week culture stimulation, tetramer positive cells here, they have expanded. But in the presence of Treg, this expansion is suppressed. So now you can compare the expanded the antigen-specific T cells versus non-expanding population. And then, CFC labeling, this population, they proliferated. And then this population, they don't proliferate. And then importantly, after resting and they re-stimulate fraction A, now they proliferate again. But this peak B, they don't show proliferation. And then they don't produce interferon gamma, TNF alpha, or IL-2. But then fraction A, they can produce these inflammatory cytokines. So peak B, they look like analgic. Well, definition of allergy is the non-stimulation upon TCR stimulation, and then no production of IL-2. And so that is the operational definition of allergy. And then they are indeed allergy. And then importantly, if you compare the staining intensity of tetramer, so here you see the tetramer high population and low population. And here not many high population. So A and B, and so the ordinate is and the tetramer staining. And you see A is higher than the B. But then TCR expression level or CD8 expression is similar, meaning that the peak A, they express in the high affinity meringue A specific TCR. On the other hand, the peak B, they show low affinity TCRs. So these findings all together, and now our current understanding is that Treg control the CD80 expression and then remove the co-stimulation to responder T cells, signal 2. And also they upregulate free PD-L1 and then interact with PD-1 and then they inhibit the PD-1 positive, inhibit the function of PD-1 positive effector T cells. And then the IL-2 receptor, the high affinity IL-2 receptor, they can absorb IL-2 from the surroundings as cytokine sink. And then chronically activated Treg, they also produce IL-10 TGF beta, IL-35, adenosine. And then most importantly, IL-10, the important function of IL-10 is the inhibits the maturation of DCs and then also contribute to downregulation of CD80-86. And then as a result, responder T cells, when they have a high affinity IL-2 receptor to peptides MSC, without co-stimulation, they'll die by apoptosis. If it's intermediate, they become anergy. And then if it's low affinity, they stay dormant. So what is important is just transient interaction with APC between Treg and APC, and then the responder T cells, their cell fate can be determined. Well, they not mere suppression, but then they may die or they may become anergic for long term. So that is good for inducing stable immune tolerance by just transient interaction with the antigen-presenting cells. So let me move on to human autoimmune disease based on these findings in mice and humans. So after discovery of IPEX syndrome and FOXP3, so really it was it's good evidence that Tregs are involved in human autoimmune disease. But how about polygenic common autoimmune diseases, which afflict 5% of the population? So since then, the clinical immunologists have looked at, examined the autoimmune disease patient, whether any anomalies in Treg function, number or function. But then result was kind of tantalizing for immunologists. And so because we cannot differentiate the cause or consequence of the anomalies, whether some anomalies it is really causative or the result. But then at the same time, we now know that an autoimmune single nucleotide polymorphism, SNPs, found by genome-wide association studies. We know that many autoimmune SNPs, and then a question is whether these autoimmune SNPs really affect Treg development or function. So that is the next question. So to address this question, the one clue is Treg-specific super enhancers. So it has been shown that in the genome, there are many sites where you see dense accumulation of transcription factors and histone activation, modification, and also CBP or P300 binding. So a very active enhancer site. It is called super enhancer. And super enhancers strongly control the downstream genes, and they are important in the determining cell identity or cell fate of so muscle cells or nerve cells and so on. So question is, regulatory T cells, do they have a Treg-specific super enhancers? And then these super enhancers, they control the Treg-specific gene transcription, and then also epigenetic modifications. And as a result, they produce determine Treg cell fate and functional maintenance as a cell lineage. So here, Treg and the conventional T cells. This is H3K27 acetylation chip sequences, and you see here conventional CD28 and CD84 locus in conventional T cells. CD28 locus highly activated, and this is called and also in Treg, they are called can be called common super enhancers. But the CTLA4 not activated in conventional T cells, but highly activated in Treg. It is called Treg specific super enhancer. And these small enhancers, specific Treg specific ones, they called the typical enhancer. So definition of super enhancers, they're putting the enhancers to small to large, and then the ordinated H3K27 acetylation, the chip sequencing, the signal level. And then you see all of a sudden, the signal become intense. This part, they are operationally defined as super enhancers. So this tangent is more than one, then the fraction is called super enhancers. And importantly, we found that the Treg specific super enhancer 66, a common one 318, and then Treg specific one, they really include the CTLA4 associated with CTLA4 gene, FOXP3, Helios, and then the CD25 IL-2 receptor, so-called Treg signature genes. And then, Treg in the thymus, they double positive to immature CD4 single positive, and the pre-Treg, they can be defined as CD25 positive, but then FOXP3 negative. And then they become mature to T-Treg, FOXP3 positive and CD25 positive. And from that stage, you see FOXP3 expression and then DNA Treg specific DNA demethylation. And here we defined the Treg specific super enhancer as the FOXP3 gene locus. And then importantly, from already double positive stage, you see H3K20 histone, H3K27 acetylation. This is the marker for active enhancer. So double positive to immature CD4 single positive, so Treg. So along Treg differentiation in the thymus, it's become stronger. And similarly, H3K4 monomethylation. This is the marker for poised enhancer. Already it started from double positive stage, and then become stronger along Treg differentiation. So these changes of the pattern, it suggests that dynamically histone needs become modified along the Treg differentiation. And ATAC sequencing, this is a marker for open chromatin. So at double positive stage, it become open and it become intensified along the Treg differentiation. So this means that before FOXP3 induction, already Treg type super enhancers and Treg specific super enhancers, FOXP3 already start to become activated, and this happens in parallel in other Treg signature genes. So FOXP3 gene locus and CTLA4 and Helios and CD25, and similarly the chromatin remodeling. So this means that Treg specific super enhancers become activated in parallel at Treg signature genes, including FOXP3. So our current understanding is that the developing T cells and with some perhaps TCR affinity for cell peptide MSC, then the Treg cell lineage become determined, and then Treg specific super enhancers become activated, and then FOXP3 induced, but then independently Treg type epigenome. So super enhancers defined by histone modification. So not only histone modification, but the Treg type DNA hypomethylation and chromatin remodeling happens. And these two together for the functional, contribute to the formation of functional Tregs. Now in humans, so we looked at these super enhancers in Treg specific super enhancers in humans. So this is the peripheral blood. The thymus produces naive Treg, and then they are present in the circulation. When they become activated, they become effector Treg. So these are conventional T cells, the activated or memory T comps or naive T comps. And then if you look at the Treg specific super enhancers, we found 21. In mice, we found 66, but then it depends on the definition. And so anyway, not many super enhancers. And then we looked for where the autoimmune SNPs they locate. And then we found that specifically, very selectively, Treg specific super enhancers these site the many autoimmune SNPs locate in the Treg specific super enhancer site. For example, vitiligo, autoimmune disease in skin, Graves' disease, multiple sclerosis, type 1 diabetes, they all the SNPs, autoimmune SNPs are enriched at the Treg specific super enhancers. So here, FOXP3 locus, super enhancers, purple, and the Treg specific the demethylation site. Well, we are very much interested in the Treg specific DNA hypomethylation because the Tregs are, as I said, highly proliferative, and then they are produced by the thymus, and then they keep maintain their function to the periphery, meaning that by proliferation, after proliferation, their phenotype and function must be kept. So among epigenome, the most faithfully inherited event is the hypomethylation, DNA hypomethylation. So we have been looking at the Treg specific demethylation. So here, a demethylation site, they are included in the Treg specific super enhancers, also at CD25 locus. And then, so they are enriched at Treg specific super enhancers, and especially the naive specific Treg demethylation sites. And then second one is activated effector Treg specific the Treg demethylation, DNA demethylation sites. So the autoimmune SNPs they can really control Treg development and perhaps function. Now, let me talk about CTLA4 and CD25. So this is the article published in 2007. Actually, this is the first the large cohort study of autoimmune SNPs and then type 1 diabetes. Not only HLA or insulin locus, here CTLA4 and IL-2 receptor alpha chain, CD25. Then these SNPs, but then whether these SNPs control effector T cells or regulatory T cells, because both molecules are expressed by both populations. So as a gain of function of gain of function variant for effector T cells or loss of function variant for regulatory T cells, that is the question. So here, CTLA4 locus, and then we found that so fraction 1 and 2, fraction 1 is naive Treg, this fraction 2 is effector Treg. And then we found that the super enhancers, and then Treg specific demethylation site here. And group 2 means activated T cell specific demethylation site here. So they are different. And then autoimmune SNPs here, rheumatoid arthritis. So thanks to the 1000 gene genome project, we know every SNPs in the genome, and then we can now define as a group define the causative SNPs. So this one is a GBAS SNP, but then perhaps the causative SNPs locate around here, and then it correlates with this region, and then Graves' disease here. So this means that perhaps the Treg specific site polymorphism here at enhancer site may contribute to rheumatoid arthritis and development. How about the CD25 locus? That here we found a Treg specific demethylation site. The purple one is the super enhancers, and many SNPs just locate this region, meaning that perhaps these SNPs control Treg function and contribute to the susceptibility, genetic susceptibility to Treg. So all findings together, the Treg pathways, mutations of Treg signature genes such as FOXP3, CD25, CTLA4 cause monogenic autoimmune disease by affecting Treg development and function. So they are now called the Treg pathways. And then about common autoimmune disease, we now know that the many the autoimmune SNPs affects they locate the Treg signature genes, and they contribute to genetic susceptibility, and perhaps by controlling function and development of endogenous naive Treg population. So, well, just a bit of a digression. This is the article published by Jean-François Bach, an eminent French immunologist. He published in 2002 that in these 50 years in developed countries, the incidence of infectious diseases they really reduced. But on the other hand, some diseases they become increased. So including autoimmune diseases such as multiple sclerosis or type 1 diabetes, and allergy, asthma, or Crohn's disease, the IBD. So just as shown in IPIC syndrome, these diseases they can develop as a result of Treg anomalies, and perhaps these people, they are genetically susceptible, and then depending on the environment, they developed autoimmune disease. That is one interpretation. So here, the genetic environmental factors can affect the Treg. And then, as I said in the beginning, that neonatal thymectomy system, depending on the genetic background, you'll see different autoimmune diseases. And perhaps these patterns or repertoire may be determined by MSC and then non-MSC genes. So type 1 diabetes, by similarly equivalently removing Treg, one individual may develop type 1 diabetes and some others develop other autoimmune disease or no autoimmunity. So if genetically susceptible and then Tregs have some the variations, and then the autoimmune T cells or self-reactive T cells may accumulate, and then so they become more susceptible. And then some environmental insult, stimulating innate immunity. Now these people develop an overt autoimmune disease. That is one hypothesis, one in interpretation of the disease. So now, Kazuyo, we have talked about too much, and then
okay.
Maybe after 10 minutes, but
10 minutes. So would you like to stop or use 10 minutes for your talk, whichever?
Okay, let me go then 10 minutes.
Okay, right. Please.
Okay. So the last part is then the treatment, Treg-based therapy. So now the natural Treg, how we can expand them to treat, to make a Treg dominant state to treat autoimmune disease. So various attempts low dose IL-2 because Treg express a high affinity IL-2 receptor. So they are very much sensitive to low dose IL-2, and then you can selectively expand Treg by low dose IL-2. And so that is one way. And second approach is they take Tregs out from the circulation and expand them by antigen stimulation in the presence of IL-2, and then transfer back to the patient. Or CAR construct put into Treg, natural Treg, and then they have some specificity, inflammation specificity, and then transfer back to the patient. A third approach is effector Treg, how they can be converted to Treg in vivo to P-Treg. And then fourth approach is ex vivo, how we can convert effector T cells into antigen-specific induced Treg and then transfer back to the patients. So in this case, not only FOXP3 induction, but also we must install Treg-specific epigenome. Otherwise, they are unstable. So by these approaches, we can expect to make the Treg, to prepare a Treg dominant state for establishing self-tolerance. So our approach is the fourth one, third one and fourth one. How we can convert antigen-specific effector T cells into Treg. So we looked for the FOXP3 inducing substance and reagents in vitro. And then we found that for this compound, this is artificial one, is 2863619, abbreviated as AS compound. So naive T cells, when they are stimulated with the anti-CD3 and CD28 in the presence of IL-2 and TGF beta, you can induce the FOXP3 positive cells. Also, without TGF beta, neutralizing TGF beta, you can induce by AS FOXP3 positive cells. But here, effector memory population, the CD44 high, CD62L low population, you cannot induce the Treg by TGF beta, but AS can induce. And then TGF beta and AS together now 40% become Treg from effector memory population. And also the CD8 positive Treg, T cells, conventional T cells can be produced to express FOXP3. And this conversion by AS compound can be achieved in the condition of TH1 or TH2 or TH17. So interferon gamma producing cells reduce, but FOXP3 positive cells now increase. Similarly, TH17 condition, TH17 forming cells reduced, and FOXP3 positive cells on the other hand increased. So also TH9 condition. And then this ACE compound, it binds to the CDK819, which is the regulatory module of a mediator complex. And so the CDK819 knockdown, you can induce FOXP3, and then kinase CDK8 or CDK19 expression. And this is kind of a dominant negative the interaction. So either one, if you express kinase and then you can induce FOXP3 positive cells. And it binds in the presence of AS, the STAT5 bind to the FOXP3 enhancer region, especially CNS0 region, and then they increase the FOXP3 expression. So in activated T cells, usually they don't express FOXP3, but sometimes in humans, just activation can induce FOXP3. And then still we don't know yet how activated just T cell activation can induce transiently FOXP3. But one interpretation is TCR stimulation activates CDK819, and then the inhibiting STAT5 activation. And then CDK819 inhibition now activates STAT5, and then the FOXP3 transcription is enhanced. So here, DO1110 lung minus mice, so only expressing the OVA peptide specific transgenic TCR. So vehicle AS alone had no change in vivo, but the OVA plus AS, now you can induce the FOXP3 positive cells. So antigen stimulation plus in the presence of AS, now you can induce antigen-specific Treg. They are very similar to natural Treg, and importantly, these in vivo induced P-Treg, they show Treg type epigenome like natural Treg. And then this is skin contact hypersensitivity by treatment with AS, orally treated. And then you can prevent the skin hypersensitivity as shown here. And then you should remove Treg, this effect is gone. In NOD mice, which spontaneously develop type 1 diabetes, just for limited period treated with AS compound orally, then you can prevent the diabetes development. And this is histological score. So their majority of the islets are histologically intact. So all these findings together, not only natural Treg or iTreg or P-Treg, now we can convert effector memory, antigen-specific effector memory T cell to FOXP3 positive Treg. We call them the stable and functional iTreg. And then they can, so effector T cells, now they convert it to Treg. And then they can suppress, say, autoimmunity or transplantation or allergy. So in vivo, it can be done, but also in vitro, AS can induce FOXP3, and also we can, I don't have time to talk about this, but we can establish Treg type epigenome by controlling co-stimulation. So these find, well, many students and the postdoc and my colleagues, staff scientists, and contributed to these works in these years. And this is a list of the people involved, and also the collaboration with people in other institutes in Japan, also abroad. Thank you very much for your attention.
Hey, thank you very much for your excellent talk. And could you turn off your slide? And so today I really learned a lot, as I did not know how the importance of Treg was demonstrated in the past, and how the research in this field has been developed. So your talk will definitely inspire many young researchers and contribute to the further development of this field. So thank you again for your talk. And before closing, I would like to share one slide with you. Let me see. Okay. So if you want to make a question to Shimon, please do it through Twitter. Please search for the account Global IMMUNOTALK and find a tweet that says, "Ask question for Dr. Shimon Sakaguchi here." And please note that due to the time difference, there may be a slight time lag in Shimon's replay. So that's all for today, and thank you for joining us at Global IMMUNOTALK. Have a nice day or have a nice evening. Thank you very much.